At Manly and on the beach, and it's a low energy soft sediment shore, and we'll just go and have a look at the compass so you've got an idea. The wind is blowing into my face, which is from that direction, which is southeast, which are the predominant trade winds here. So this shore is really sheltered by that rock wall. Just shoot a bit of this. If you can see that. There's the shore, and there's basically southeast in that direction. So really, really well sheltered shore. Artificial. Council put the sand here, so it's not really representative of the sand that's around here. And if you can see far out there, there's some people having a wander around on the shore, and the tide is round about low now. So we've got a mix of habitats here. We've got some sand. We then run down to some sort of rough rubble, and then there's some rock exposure as well. And on the right, which is to the south somewhat, there is some mangroves. So it's got quite a range of habitat types. But the sort of sediment we get here, this is artificial stuff. This is the sand. This sand is, of course, brought here, so it's not part of the natural habitat, but it's starting to mix in to the other parts of the beach here. You can see the tracks here. This is from the council having been down to remove strands of cyanobacteria from the shore. There was a big pile of cyanobacteria here a few weeks ago, and they've now cleared that, and that's what the track marks are. So this shore is a mixed shore. Okay, so the sediments will be very complex. It's not like a sandy beach you get when Noosa or wherever. The sort of sediment beach you get here is very mixed sediments. So you get some very fine sediments, coarse sediments. If you see, there's lots of shell fragments you can see lying around. There's also seagrass fragments as well, and that's because it's mixed energy. So today, with the wind blowing from sort of east-southeast sort of, then what we've got here is a shore where the fetch is hidden by the seawall in front of us. So basically the only distance waves can set up is over that very short distance just from the seawall to here. Now if the wind would be coming from further to the north, then you would expect a much longer fetch, and therefore the wave setup could be larger. So depending on wind direction, it really drives the energy that this shore experiences, and it's a mixed energy shore. Hang on, I can see a toy. Is it? All right, okay. All right, so anyway. Yeah, so here we're on basically, it's pretty much sand. It's come from the beach, just been moved down the beach over the years, and you can see that the sand here is fairly well sorted. There's a stingray hole, so there's a bit of vertebrate bioturbation, and if you look at the crab burrows, this is soldier crab feeding marks. There's a bit of animal bioturbation as well. And indeed, Scopamira, so that little hole there has a little crab in it that makes those balls. So they feed in a similar way to the soldier crabs. I'm here with my good friend and colleague Dave Brewer, Brewer Consulting. So if you want to sign up any contracts or get a job, come and see him.
No thanks.
What do you mean, no thanks? He's a scruffy bugger, but he does do well. He does do well. All right. So apart from the swallows, we're helping out the beach. So I just want to talk a little bit about the water and sediment. All right. So I mentioned that wave action here is going to be mostly very low because of the shelter that's provided by the seawall. So that's going to mean that the water velocities coming over here, the amount of energy that's delivered to the shore, is relatively low as well. So there's a relationship, a thing called the Hillstrom diagram, which you may have heard about in class, and that says that as you increase the amount of energy, the current velocity in a system, so the water can pick up, suspend, or erode rather, larger and larger particles. And then those particles can remain in suspension until the water velocity decreases, and then as the water velocity decreases, so they start to fall out, the heaviest first, and then so on and so forth. So this is used by a range of animals. So we're going to have a look. You can see the soldier crab burrows here. We can't see the soldier crabs. That's probably another section of video. At the moment, the soldier crabs are actually working from underneath the sand to to get food. They get detritus from the sediment by using the same principle of basically flotation. They basically move the sand rapidly with moisture in their mouth parts, and that floats off the lighter particles, which are detritus, basically detritus and bacteria and algae and those sorts of things, leaving the sand behind and then dumping it, which all little balls are. And that's what, here's another Scopimira, and that's the same thing Scopimira does. So if soldier crabs do this, Scopimira does it. And so there's this relationship between moving water around and sorting sediments. And the same principle goes even if you go up a scale. We go to stingray feeding pits. So we'll walk over here, and here's a couple of stingray feeding pits. You can see the outline of the stingray. I'm trying to find a good one here. What's the best? Oh, this is pretty good. I'll find another stingray feeding pit. Got to have a good one. It's a little one. Damn it. There's no good perfect one. Anyway, I'm going to cut in that last one. Anyway, what the stingrays do when they come in, we'll ignore the bird footprint. What the stingrays do when they come in is they'll use their ampulla lawrencini to identify something. That's the detect electrical charges in the water. It'll detect that electrical field generated by the animal living in the sediment, and then the stingray will dig. And the way it digs is it draws in water through its spiracles on its back and just behind the eyes. Water gets forced through the gills, out through the gills, and then the ray is flapping its wings, and that action generates a current which lifts sediment, erodes sediment, and then the sediment's placed to the sides. So that sediment is placed to the sides of the hull, and then it's using the same principle, energy into the system, and you sort the sediment, you move it around. Now, of course, in a shore like this, where you've got lots of mixed sediment, you've got pretty low energies, you're going to get a lot of detritus around the place, and you can see the detritus in that burrow. That's basically seagrass fragments that are breaking down, and that detritus is going to become incorporated into the sediment. So the sediment's going to be nutrient rich, whereas on an exposed sandy shore or sediment shore, there's going to be a lot of high energy waves, and those high energy waves are going to do the same thing. They're going to sort the sediment. They're going to lift all of the detrital particles out, or most of the detrital particles out, and the sand will sort according to the wave energy, and that's why you tend to find very uniform sand on sandy beaches. Whereas a beach like this, we can move from sand here. There's some pebbles around the place, and over here we have some gravel. I'm actually walking through a different sort of sediment now, so I've gone from that nice sand now to basically muddy sand. All right, muddy sand is a bit squidgy. It's got lots of sand in it still. And now what we're going to do is we're going to do a little demonstration about energy into the system and then what it does in terms of particle sorting. So I'll just pause a second. So there's the jar. We've just added a bunch of energy, and we'll just set it down and come back to it in a little while. So we'll see what happens. But I was mentioning the Hillstrom diagram earlier on. So in one direction you've got erosion, a certain velocity particles will erode, and at another velocity, and then at a different velocity, larger particles, at a higher velocity, larger particles will erode. Now when we reverse that and we lower water velocity, then what we're going to see is that particles are going to start to fall out, and as we'll see hopefully with the jar demonstration, they'll fall out in sequence of their size, roughly, and maybe we've got some detritus material sitting on the top, which illustrates the point about the way these animals on the shore here feed. The other things that can slow water velocity are structures. So where you have complex reef habitat, here we've got some rocky shore that we'll do as an analog. You can see the water passing over that will slow down, particles will fall out. Where you've got a burrow or a hole such as those stingray holes that were dug over there, that we saw it back there, then what's going to happen is the water velocity will drop across as that water passes over that burrow. So particles will fall out, and you saw that in terms of the detritus in the bottom of that burrow. Much more detritus in there than there was either side. So down here, we're just moving across to a mangrove area. These are Avicennia marina. No doubt someone will have told you about these in class. You can tell they're Avicennia because I just slipped. You can tell they're Avicennia because they've got lots of pneumatophores, right? Those peg-like things sticking out of the ground. And you'll understand that those will act to lower water velocity again, a complex structure, and then that will cause particles to fall out and trap other particles. Here we see that there's a couple of mangrove shoots here, young ones growing up, and you can see around them this black stuff that looks like, what does it look like, Dave? Well, it is, yeah. I meant in sort of an analogy. It looks like somebody's like coughed something up over it. But this is actually Lingbia majuscula. It's a cyanobacterium, and it's been causing a bunch of problems in the bay, and it's doing really well. But here you can see the roots of it, the pneumatophore roots of the mangroves have actually trapped amounts of this stuff. And there's a story about that. Sorry?
Tents of Lingbia.
Yeah, there's a little Lingbia tents scattered around the shore, and that's an interesting phenomenon. So now I slipped over just now because we're actually moving into mud. So mud, smelly mud around mangroves, very important stuff. You can probably see get an impression from the green in the pools here of the amount of photosynthesis going on in some of these pools. These are probably Lingbia mats again. Lingbia cyanobacteria tend to be bright green. Diatoms, which are very important primary producers on shores and on the surface of muds. I mean, diatoms are really, really important. And those diatoms generally have the appearance of like an olive drab colour. So if you see bright green, or conversely this black, this is a Lingbia breaking down, then that usually suggests it's a cyanobacterial mat as opposed to a diatom mat. You know, diatom mats are pretty interesting things. They're interesting partly because, you know, diatoms can move. So the pennate diatoms, which dominate the diatoms in shores like this, are actually able to move. So they're photosynthesizers that can move around. So they move in and out of the sediment, often with the tide. So you can see lots of primary production. There's primary production being done by the cyanobacteria here. There'll be diatoms in there as well. We've got the mangroves doing it at the back, producing a lot of material which then drops down. And so the muds around mangroves are going to be very, very nutrient rich. There's not in this low energy area. So if we have a look, let's see what I'm seeing here. Oh, this is pretty harder than I thought. If we have a look and turn it over, maybe you can see there that mud is basically, it's got a lot of gravel in it as well, but that material is basically a very, very thin layer of brown on the surface and then dark below that. So that dark stuff, where you'd smell it, you'd smell hydrogen sulfide. So the surface layer is oxygenated, and the bottom layers, lots and lots of nutrients. The nutrients are being used up by bacteria. The bacteria use up all the oxygen, and then the community of bacteria moves to a sulfur-reducing bacteria. And those sulfur-reducing bacteria give off hydrogen sulfide and a whole range of other things. And that forces any of the metazoans, any of the more complex life forms, to move out into this oxygenated layer. So the active sort of living things, the non-bacterial, are just confined to that few millimeters on the surface there. So that's mud, and so the stuff that lives there can only occur in that upper few millimeters of oxygenated habitat. If we move to an area where there's been some bioturbation, we'll move back over here. Sorry, Dave. If we move back over here, see Lingbia patches around the place, and we'll find, oh dear, the girls are, sorry, better not start recording that. Here's another area, lots and lots of mixed sediment. You can see some red on the surface. Here's another Scopimira been working, the little balls here. And see some red in the surface sediments as well. I'm pretty sure that's iron, and iron of course is an important pigment in photosynthesis. Here's another section of shore, so we just moved up a little bit, no bioturbation, and look at the black layer there. Okay, look at the black layer there. It's really difficult to see with this. So there you go. So sediment very stable, not much bioturbation at all, and therefore we get a very strong, what's called a redox discontinuity layer. The difference between the black sediment and that layer of brown over the surface. You can see where the Scopimira been feeding. The balls are black. Right? That's because it's bringing that black material, the reduction zone material, out onto the surface of the sediment. So it'll become oxygenated, and then it will. Yeah, it's challenging, isn't it? I just can't see anywhere. You're right. Can you see anything? No, it's okay. It's still going. Yeah. Okay. No, I can. All right. That's good. Okay, I'll do it without glasses. Okay. So anyway, very, very sharp zone between the oxygenated sediment and the unoxygenated sediment, and that's demonstrated by the Scopimira balls, which are black, and they're black because the Scopimira, as they feed and pull sediment out, they're bringing the reduced sediment up to the surface, the black stuff. It's then going to get oxygenated, and those nutrients are going to circulate in the system. Let's find another more disturbed area. Let's go back to the soldier crabs. Where are the little soldier crabs gone? Oh, they're Dave. I've lost the bastards. There they are. Girls are enthusiastic, aren't they?
Amazing morning.
Yeah.
Beautiful morning.
Lovely, isn't it? So here we are back at the soldier crab zone. We'll have a look at the redox layer here. Now, if you can see, if I look, if you can see there, you'll see that there's no sharp division. There's darker sediment deeper down, that's what we'd expect, and then it gradually grades into yellowish oxygenated sediment. Now this of course is coarser material, so water in a coarse material will percolate through rapidly. Dave, could you assist with a demonstration? Pour a bit of water on the sand, on just there please. Okay, stop. Where will the water go? Okay, so that water's passed rapidly through the sediment, and that's going to be carrying oxygen with it. It's all right. So this sort of sediment, we've got sand basically, the water will percolate through it very quickly and carry oxygen deeper down. If we go to a muddier area, we'll try the same thing. Come back to our jar in a minute. There's a muddy area. What does it possibly do us? Same thing here, Dave, if you wouldn't mind. Give it a go.
So mud just set up.
Yeah, hang on, let's have a look. Yep, lovely. How's it going? Dear. Dear, it's not sinking in. It's not sinking in. Couple of reasons here. You can see the green on the surface. Again, that's probably cyanobacteria. It looks a bit light to me for diatoms. So, and because there's no bioturbation here, this is a much. We go back and have another little diggle in here again, just to remind us about that very thin layer of oxygenated sediment on the surface. So very little bioturbation here. It's a coarser sediment, lots of nutrients, and water is not percolating through, so it's not going to carry oxygen deep into the sediment. There's lots of...
So this is a bound layer on the top?
Yeah, it's like a bound layer on the top. So one of the important things that bioturbators do, things like soldier crabs and Scopimira and stingrays and bêche de mer, sea cucumbers, very important in this regard. What happens is a mat forms on the surface, and that mat is basically seals off the sediment. That stops the oxygenation of the deeper sediments, and if they're nutrient laden, then they'll build up lots and lots and lots of nutrients in a reducing environment, and then when that's released, that's when you get things like acid sulfate soils, which someone will talk to you about another day. So yeah, interesting stuff. Well, let's go back and have a look at our jar. So all this we've been talking about now is about the energy environment on the shore and how that impacts the sediment, and then the nutrient relationships which are impacted by the animals. All right, Dave, how's the jar going, mate? Can I give you that a second? Let's have a look. There's our jar. So our jar, if you can see, has a layer of, well, it's got some water on the top of it, which is a couple of inches of water there, and then down at the bottom we've got coarse material. There we go. Down at the bottom we've got coarse material. Thanks, Dave. And then right at the very bottom would be the coarsest stuff. And then we've got a layer sitting on top of the sediment, which has, that layer has sort of, if I just shake the jar gently, you'll see it's still somewhat suspended. Somewhat looks like a gel on the surface. So those are the very fine particles, and detrital material will be in that as well. But in the water, here, I should look at the jar and look through the lens. The water that's sitting on the surface there, some particles haven't fallen out, and that's because they're so fine, they really need either a long time, or they need some agent other than just physics to sediment them out. Things like filter feeders will sort that material, and a whole range of other organisms will be able to process that and then filter those particles out. So yeah, interesting sort of job. You see the general principle: energy in, and then that separates out the particles. If this was a very high energy shore, like Noosa or off the shore at Stratty, then of course all those fine particles would remain in suspension. All the detrital particles would remain in suspension and then be carried away. And that's why those beaches are so clean. And if you dig down in those beaches, you will not find a black zone at all. The water percolates through really well, and they do very nicely. All right, so the next question we're going to address is what sort of habitats are available for living things on a shore like this. Now, obviously, there's the surface. All right, we've identified the surface. We've identified some living cyanobacteria, diatoms that will live right on the very surface. We've identified that the sediment can be stratified in terms of its oxygen status. All right, so that black stuff, nothing other than those bacteria can live in there. All right, so everything is driven up towards the surface. I've got a little graphic that I can show you in a bit about that, but basically living things on this shore are then confined to that area above that deoxygenated layer. So obviously where we were higher up on the beach and bioturbation was taking place, there was the ability for organisms to live down deep in the sediment, relatively deep in the sediment, whereas here on the shore where we've got these mats, you saw evidence of life because we showed you some Scopimera, and they were excavating the black zone. So things that can make a tube or a burrow can live in those sorts of sediments, but then you've got to be a big thing that make and maintain a tube. Something like a large polychaete worm, for example. Some of the sedent polychaetes do that very well. Some of the large clams can do that, and these creatures can live in these zones where the sediment's fairly stable, and they can live quite far down in it as long as they can maintain a burrow and then draw water in through that burrow to oxygenate that, to obtain oxygen through their gills. So surface areas are quite important. What have we seen that's alive on the surface so far? Not a lot, but if we look around we'll probably find some things. I think over here, this shellbrit stuff's quite important, quite interesting. So here's a shell. You have a look there. So that's Pyrarsus ebeninus, that's a mudwhelk. And mudwhelks crawl around the surface of the sediment, feeding on those surface nutrients. All right, so they'll be feeding on the diatom mats. I don't know whether they consume the lingua or not, but they certainly. So there's organisms that can creep around the shore and basically living on that very rich nutrient laden surface sediments. So the surface of habitat, there's some more pyrazis here. Right, so the pyrazis density is generally very high. So another place you can live, so there's the surface, or you can make a burrow, and the other option is a pool. Now we're moving back towards the pool we filmed earlier, and if you can see, there's a lot of oxygen generation going on. There's bubbles everywhere, and that pool is going to be a very oxygen-rich environment during the day, at least. And a pool provides a little safety bubble, if you like, for organisms that might stay while the tide is out. And then, of course, we're visiting the shore here at low tide. So pools such as this are important areas for some animals. Pools can also be generated by things like stingrays. So stingrays dig the pits I showed you earlier. The pits we saw there were dry, but in many beaches those stingray pits will actually retain water, and then they become a habitat for organisms as well. And we've done some research a few years ago now that showed that baby whiting and juvenile king prawn recruit to those habitats. So the stingray pools and pools such as this can be very important for creatures such as that. We'll see if we can find some baby fish. If your eyes are good, you might see fish moving around. Those are actually gobies moving around. There's a little goby over there. You can tell the gobies. There's Dave's shadow. So, hey Dave. Oh, he's chasing one. He's chasing one. That one's not a goby. That's a baby whiting. All right. Now that we know it's a baby whiting because it's not burrowing in the sediment. There you go. It's a juvenile whiting. You can see it's tiny. There's also here a pond skater. All right. That's Halibates. Pond skater. If I'm catching any of this, Halibates is a pond skater, and they sit on the. They basically stride the water tension. And there's also this freshwater equivalents called hydrobates. Oh, actually Dave's found a hot spot. We've got a hot spot of baby whiting. We know they're whiting because they're not burrowing in the sediment. If you see gobies, then they go crazy. They're mad. And they get a great advantage from this because several things, if you think about it, if they're up here in the pool, they're not down at low tide getting eaten. Actually, these aren't. Some of these terrapon jabua, I think. What are they, striped grunter? What are they called?
Yeah, they're grunter.
Little grunter. They're striped grunter, yeah, maybe. So, settlement habitat for a number of species. So, several benefits. Benefit one, lots of nutrients. Benefit two, well, oh, Dave, yeah, Necarius. Dave's just pointing at Necarius. That's a whelp. It's a predatory whelp. It'll feed on carrion as well. So that's Necarius. It's got a siphon out the front. Oh, he's rolling over. What's he rolling for? I don't know. Anyway, whatever. Maybe it hates the lingbeat. Anyway, the siphon, it's smelling around the habitat, and it's trying to find some food to feed on. And generally, the whelks are either predators or carrion feeders, and they respond quite well to dead bits of fish and things like that. So anyway, so there's a thing. So we've seen fish there. So the other benefit, going back to the fish, the other benefit for the fish. What's that, Dave? There's some little cerithids in there, some more snails. They're not moving around very much. They seem fairly subdued. Well, we normally expect things to be fairly active at low water, because that's when all the nasty predators, the ones that live low tide, occur. So back to the pools and the values of fish. So one is lots of food, which is terrific. So lots and lots of food. They can feed while the tide is out, which is nice. They avoid predators, which is good. All right. So predators can't live in this habitat when the tide's out. So they get a spell of some 6 hours or so where there's no predation, and they can feed fairly well unchallenged, apart from people like me walking through puddles. And then also the third benefit is probably thermal. So little fish need to get to be big fish, and they need to grow fast to escape predation. All right. So size gives you an advantage in the predation wars. You're faster, you can access more habitat, so it really gives you a leg up, and also you can avoid a whole range of predators the larger you get. So the heat in these pools, and it's a warmish day, about 29 I suppose today, something like that, but in the pools here it will be climbing well into the 30s, and that gives them a thermal advantage. They can digest food more quickly, and therefore they can grow more quickly. So pools are beneficial intertidal habitats. Another habitat here, and Kath won't forgive me if I forget the mangroves, are actually mangrove trees. So the pneumatophores provide a habitat for things to live on, things can attach to those surfaces, and then they also, there's a habitat here up in the leaves, which is often dominated by insects, but you may even find, well, there's insects dominating the habitat. You can see them flying around. I remember shooting footage of hummingbirds in the States, and then getting back, couldn't see anything. There's a bunch of insects in here, so yeah, them dropping into the water would provide a source of food for fish, no doubt. Let's try and see if there's any snails. Dave, can you see any littorinids in there? I don't see any, but in many mangrove stands there are lots of littorinid snails, periwinkles, that will live up in the branches and graze on the surface of the mangrove bark and leaves. Can't see it at the moment. Anyway, good. Anyway, so habitats. What other habitats are there, Dave? There's some rocks. Let's go and flip a rock.
Good rocks there.
Yeah, some nice rocks. There's a lone rock over here. I cut my finger last time doing this, but there's some more gravelly stuff. Yeah. That's all native rock, right? That's the stuff that was here, should be here. Here's a very lone rock. And let's have a little flip of this one. There's oysters on it. So this is a little outlier here. Lord knows how it got here, but there's a rock. There are some rock oysters, which is a very cleverly named animal. And those rock oysters are welded onto it. You can see some barnacles on the surface. And then below, nothing. Well, a little crab, but very little else. All right, this is a pretty tough environment. So this is a snail, right? So pretty tough environment. Little top shell. So if you're an animal living here, the benefit of the rock, all right, is actually shelter. So because the rock is shielding you from the sun, you're going to lose moisture less rapidly, you're going to be hidden from predators, and all of those factors make rocks, where they occur on shores like this, important.
Of course, they're a solid substrate too, for things to grab onto and hold.
Yeah, so that's right. So it's a solid substrate, and it allows animals then to fix in place and basically wait the incoming tide. What else we got? Oh yeah,
What are these?
fucking fine, mate.
Big
Oh, it's a burrow. Yeah. So the question is, these burrows here, what are they? We think possibly crab. If we stand away from it for a little while, we might see the crabs come out. It's the sort of thing a crab would make. There's a deposit pile here, maybe, and then there's the actual burrow itself. We'll turn around, keep an eye on that. So here on the shore, we're getting some other native rock. Here's another burrow, and there's another one. I can bet you that's crab. That's got to be crab.
Nice clean sides.
No, fiddler crabs probably.
Yeah, wonder about that.
Nice.
It's a different pool.
Little oyster clump. So oyster clumps are quite important as well. So there's a little rock. The oysters will weld onto the rock, and then oysters will start welding onto oysters. Before you know it, you've got several kilos of filter feeder hanging on those rocks, and of course they're going to be doing a job filtering the water. Indeed, if I just scan over here a little bit, you'll see in the distance there that those white patches in front of those mangroves. Those white patches are basically little oyster clumps, and all those oysters, when the tides are in, they're feeding, they're filter feeding, and they're removing fine particles from the water, and then either depositing them as pseudofeces. That means they sort them out in their filtering system in their gills, or those their food, so they take them in and then add to their biomass. So here we get onto real rock, if you like. You can see here that actually interestingly, and it's dried out now, which is good, but this is more of that Lingbia stuff, that blue-green algal mat that we showed you earlier. So where sediments come in and lay on the shore, then basically what happens is unless they're removed by an animal, by something like a grazer, then basically they settle on the surface and form this crust on the surface. Of course, that's going to smother anything that might want to otherwise live on that habitat, so not so good. And here we get to down the shore. I don't know if you've noticed, we came from a zone where there are no oysters and now we're in a zone where there are oysters, and basically as we go down the shore we're going to find probably a greater diversity of organisms because they're exposed to the air for far less time. Here's another little pool and see some fish darting around. If you see a fish dart and then stop incredibly suddenly and then disappear, it means it's a goby. All right. Everything else you can generally catch sight of and identify as they carry on moving. Here's a nice little oyster outcrop. It's doing very, very well, and that oyster outcrop is going to be doing great service to the bay in terms of filtering the water. So it's important we follow the new rules and not remove shellfish from the shore. You're still allowed to eat them in place. You might see a few valves scattered around where folk might have come down and helped themselves to a bit of a feed off the rocks here. Going lower down, getting down towards low water now. More fish. Oh, we got Padina. All right, so when we're on the upper shore, we're talking largely about microalgae. All right, we're talking about. We were talking about diatoms, we were talking about cyanobacteria. I didn't mention much about dinoflagellates, but they're there as well. But down here, as we get lower on the shore, so conditions are more marine, less time is exposed to the air, and then what we tend to find is we get algae. And here we've got some macroalgae, and in front of us is an algae called Padina. Those of you who've been to the reef may have seen it, sometimes known as sea trumpet, I think. And Padina is pretty important because it holds calcium carbonate as part of its protection. It's a brown alga, and when it breaks down, then of course those fine particles of calcium carbonate go into the environment. There's patches of it around. There's some other algae there, I'm not sure, but probably again Lingbia, fragments of seagrass. Some more Padina. More Padina. Lovely. Get your glasses on, Tibbet, so you can see what you're looking at. Ooh, I can see something. So there's more Padina, beautiful. And here we got a burrow. This is probably a crustacean burrow. You'll see it's actually, if you can see that resolution, it's actually stirring the water up. That's pumping. It's pumping water through, so it can respire, and it's living down in an anoxic zone because the sediment is black. What do they do?
I don't know.
Another hole there. Maybe living in that burrow area. I don't know. Anyway, some respiration going on. Oh, hello. There's a crab that met its maker. Well, actually not. That's just ecdysis, so it's not being cooked at all. No one's killed it.
It's a molt.
It's a molt. It's basically crawled backwards out of its shell because it needs to increase in size. It needs to grow.
Possibly lives in that hole.
Do you want to stick your finger in? No.
Big enough for her.
We'll volunteer somebody else to do that. Where are we now? We're down in the oyster zone. That's why Dave and I are wearing the appropriate PPE of sensible footwear. I lost my booties, so I don't know where they're. Oh, big fish, big fish. Where did it go? Let's have a look. Oh, loving it. There's fish everywhere here. All right. Look at that. Beautiful. There's every space is used, so that hole over there, that'll have a crab in it, you can bet your life. And there'll be crabs and shrimp all through this. And one of the things one can do, is actually introduce a bit. Dave's putting his hand in the hole, try and see if he can elicit a response from the angry animal. Doesn't, so moving on. But one of the things that you can do in a pool like this is bring down a little bit of bait, and you can pop it in the pool and have a look at what happens. And I don't know, we might do that, Dave. We've got any bait on us, have we?
No.
We're not allowed to kill shellfish. What's that, mate? Let me have a look. I'm coming. Oh! It's getting slippery. Where's this can? Oh, yeah, okay. So there's a sac pulsating. I'll just make it pulse again. Sac pulsating, and that's probably worm eggs. All right, so a number of invertebrates lay their eggs in sacs, gelatinous sacs, mucus sacs, and I think those are worm eggs. Just say wobble again, wobble, 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 wobble. All right, down we go. there's another oyster zone. So oysters, we're moving in sort of rocky, almost a rocky shore area now, and oysters, you might know, tend to occupy, these rock oysters tend to occupy about mid-shore level, roughly. And we know we've got a neap tide today. The tide didn't go out very far, and basically we're at the limit of our exploration. Oh, interesting. Just in front, we've got some fish swimming around that rock. I don't know what the res is at 7:20. Hopefully you'll be able to see it. Those are little whiting. Oh, no, it's Terrebonne jabua. It is. It's a little striped grunter. Or, what's they called? Terrebonne jabua. Not surf perch. No, I'm not going to find it. Anyway, they are Terrebonne jabua. So maybe those recruits we saw in the pool earlier weren't whiting, but certainly we have seen whiting here. And so this is the rising tide. So,
Grunter.
which one?
The one common name, crescent grunter.
Oh, crescent grunter, and there's another one over there. So, what these fish are doing, all right, these small fish, they're not in the pools, they're actually right at the very edge of the water as the water laps over the shore. What they're doing is they're having a thermal advantage, because as the water moves up the shore, so it's collecting the heat that's been absorbed by the sediment, and dark sediments contain more heat. So these fish will be living in a zone that's very shallow, it's warm. We called it the tidal excursion front in a paper a few years ago, which we did actually just down the road in Lota. And what we did was we put temperature sensors on floats and then brought them in with the tide, and it showed that the water temperature spiked really at this front edge. This is the leading edge of the flooding tide. So this small volume of water has been heated up by hot sediment, and you can see the sediments over there it's going to leak onto, and that heat exchanges, the water warms up. So we've got shallow water where there are going to be few predators for these small fish, the surf perch, crescent perch, sorry, that we've been looking at. They're going to have a thermal advantage in terms of growth. They're going to get first access to food, and so they've got the similar sorts of benefits to those fish that are living in the pools, but of course they have to be fairly robust physiologically to withstand high temperatures, low oxygen that you'd associate with this sort of water. Backing on to the sediment properties, if you get sediments can either be dilatant or thixotropic. All right, so this is a thixotropic sediment. So what that means is when you stand on it, it squidges out to the side, which is why you've got that child's imprint. You'd expect that in sort of muds, clays, that sort of stuff. When you have a saturated sediment of coarser particles, then what you'll tend to see is not that happy. You don't displace the sediment in terms of leaving squidgy footprints, but what you do glasses to it. Sorry, Jesus, I'm hopeless. Am I vain? Is that it? What that sediment does when you've got a sandy sediment that's saturated with water, when you stand on it, it basically you're putting energy into the beach, and that will squeeze the water out from between the grains, or if you do it repeatedly, it'll put energy into it so the grains will float and your feet will sink into the sand. They won't make squidgy footprints. This tide's getting in, isn't it? Dave, what have you got?
What these are in?
Oh, mulberry whelks, brilliant. So, diversity again higher. These creatures here, little things, look a bit like mulberries, and their name is mulberry whelk. The Latin name for these is Morula marginalba, I think. Morula, if you recall from embryology, is basically raspberry-like, and that's an embryological stage where there's just a ball of cells. I'm going to look underneath, I'm going to have a quick squeeze. Nothing! Oh no, not a lot.
Was a good crab.
Hopefully watch what happens. There are crabs feeding on the surface. Okay. I'll just move up. You see them run away? Hopefully you saw them run away. I should look in the camera. So when we're walking the shore, one of the things, here the crab burrows, again, it's a crab burrow, it's a fiddler crab burrow. When we're walking the shore, things are looking out for terrestrial predators. Things are looking out for terrestrial predators. And so, and particularly the crabs have very fine-tuned vision. So these fiddler crabs feeding off the sediment, fiddler crabs have one large claw. The males, sorry, have one large claw. The females have equal sized claws. The male claw is for display, and they have these fights and they control habitat and area, and all these areas here are basically crab habitat. You can see the crabs running away maybe if 72p is high enough res. So they're very good at spotting things from a distance. Crabs basically key in on movement. So if you stand very still in an area for a while, you'll see the crabs actually coming out of their holes. Yeah, they are. They're running to holes. They're running away. They're running away. It was about an hour after we first did the jar experiment, so we left it up top of the shore here, baking in the sun. And what's happened? Well, look here. Dave, perhaps I can get you to hold the bugger up. Have a little look in the sun. Where's the sun coming from? There. It's almost midday, isn't it? So if you can see in there, it's got clearer, and even finer particles have fallen onto the top of that sediment, and so we're looking good. The experiment kind of worked, and you now know the relationship between sort of energy in the water and particles. Lovely. Thanks, Dave. Bloody marvellous. Where's that toy? Oh, someone's nicked it. Alright, so as we walk back, yeah, please, mate. Oh, actually, no, oh, do I need it? Yeah, I'll take it home with me, just in case. What I need mud for at home, I have no idea. There's a, I interpret that. So what's going on there? tell me what is going on there, what's the relationship, what's doing it, what's going on. And then here, what's going on here, what's the process, what's the animal doing, and then how does that contribute to sediment structure. And then, hello, now we just walk up the shore. Anyway, so to recap, we're basically at Manly, and we've described some of the stuff you can use to interpret life on a low energy sediment shore. So tomorrow, see where we end up. But Dave thinks it might be worth having a look in the marina. I tend to agree. Pretty exciting in there. Alright, end of.